Hello, I'm Pavitra and welcome to Excelogen Biotech. Today I'm here with the topic genetic mechanism behind gonadal sex determination. So what is sex determination? Fertilization occurs, zygote is formed. This zygote will develop either into a male organism or into a female organism. So how is it determined, how that occurs? This organism have to be a male or a female. How is it decided? So we are going to talk about that, the genes which act behind this mechanism. In the early stage of an embryo contains a bipotential gonad. What does bipotential gonad means? That is, this gonad have the ability to form both a male and a female organism. So there are basically 4 transcriptional genes, 4 transcriptional factors. These factors are SF1, WT1, LHX9 and GATA4, that is GATA4. These transcriptional factors are already present in a gonad, a bipotential gonad. It is determined, the decision is taken whether the bipotential gonad must transform into a male organism or a female organism. So in the bipotential gonad there are many group of cells. So 2 branches of cells are present and one branch say make male organism and the other says don't make female organism. That is, one branch says make male gonads, B says don't make female gonads, and the opposite acts in the case of male organism. So this is how it acts. It is very accurate pathway, and pathway contains negative and positive feedback mechanisms which will prevent any error occurring. So let us move on to the pathways in detail. So moving on to the ovarian pathway, the pathway which develops ovaries. So we know already 4 transcriptional factors are present: WT1, SF1, LHX9 and GATA4. So if no Y chromosome is present, these factors will lead to the overexpression of WENT4 protein. So along with this WENT4, which is already present in small amount in the gonadal epithelium, R-spondin 1 protein, which is a small soluble protein, R-spondin 1. This this protein is also expressed. WENT4 in synergy with R-spondin protein together acts to produce beta-catenin. This beta-catenin will lead to the development of ovary, and it also blocks the action of SOX9 factor. So what is the SOX9 factor? Testis promoting transcription factor. So this is also blocked. So on one side the ovary development is promoted and on other side the testis development is stopped. This is how the action occurs. So now, if an organism with XX chromosome have R-spondin 1 gene mutation, so what will happen in that organism? So we know R-spondin 1 protein along with WENT4 protein together acts to develop beta-catenin, and this beta-catenin is the one which develops the ovary. So what will happen if there is no R-spondin 1? Eventually the XX chromosome organism will develop into a phenotypic male organism. So in a natural male organism the phenotype must be XY, but here they have XX chromosome but its phenotype is male. This is what happens. Now, what will happen if chromosome 1 is duplicated? Chromosome 1 have the region which contain R-spondin 1 and Wnt4. So what will happen? One more copy of that chromosome is present in a male. In a male organism, what will happen if the chromosome 1 area with Wnt4 and R-spondin 1 is duplicated? This organism will develop into a female. So here occurs a sex reversal from male to female. This is what happens. So if we are inducing an XY mice with overexpression of beta-catenin, this will also lead to the development of a female organism. Isn't it interesting? So we can reverse the sex of an organism. So this is how ovary is produced. This is how the signal is given to produce ovary. The testis pathway. The same set of genes in the presence of a Y chromosome will activate SRY gene, sex determining region of the Y chromosome. And this SRY gene will remain activated for few hours only. So this SRY gene, whose basic role is to activate SOX9 gene, and the later functions are done by the SOX9 gene. SOX9 gene performs many functions. The first one is it activates the development of testis. Second one is it inhibits the role of beta-catenin, which synthesizes ovary, and also it binds to cis regulatory region of testis development promoting factors, which include DMRT1, and this DMRT1's role is maintenance of testis. FGF9 have two roles. One is the development of testis, to promote the development of testis, and the second one is to maintain the transcription of SOX9 gene. And anti-Mullerian hormone. Anti-Mullerian hormone, what is the role of anti-Mullerian hormone? Anti-Mullerian hormone will degenerate the ducts which synthesize uterus. So these are the roles performed by SOX9 genes. This is how the whole pathway works. Thank you.